Good day everyone, we are here to present to you all on a medical condition which is breast cancer. So our group member includes YEAW, Changhua, Peini, ZHANG, JOSLYN and TIING.
What is breast cancer? Breast cancer is cancer that develops in breast cells. It forms in lobules or the ducts of the breast. There are two main categories of breast cancer, mainly invasive and non-invasive. The invasive ductal carcinoma begins at breast milk ducts and it invades nearby tissue in the breast, whereby the invasive lobular carcinoma develops at the breast lobules and then invade nearby tissue. The ductal carcinoma in situ and lobular carcinoma in situ are non-invasive cancer, meaning that they do not invade surrounding tissue. Breast cancer can be diagnosed via ultrasound and mammogram. The symptoms of breast cancer includes a breast lump or tissue thickening, breast pain, swelling in all or part of the breast, bloody discharge from nipple, and sudden change in shape or size of breast.
So let us move into HER2 pathway, at which HER2 pathway is one of the most prominent pathways that can lead to the breast cancer. So for the HER receptor, generally pairing up or dimerization between the HER receptor is needed to trigger the signal production, and undeniably, receptor pair that containing the HER2 will produce stronger signal as compared to other receptor combination. And HER2 positive is recognized as the most aggressive breast cancer as it can grow and metastasize rapidly and it has poor prognosis. So basically, the part that being framed in red is the PI3K, AKT, mTOR pathway in the HER2 pathway. So firstly, dimerization of the HER2 will lead to the activation of PI3K, at which the PI3K will phosphorylate the PIP2 into PIP3, thus leading to the phosphorylation of the AKT. Then this event will consequently trigger the downstream activation of GSK3, MDM2, and mTOR. And finally, cell proliferations will be provoked and the self-apoptosis will be inhibited.
The MAPK-ERK pathway is activated by the HER2 receptor. This leads to the recruitment of the growth factor receptor bound protein 2, GRB2, and the activation of guanine exchange factor, SOS, and a small GTP binding protein, RAS. Activation of RAS leads to a kinase cascade, which then activates RAF, MEK, and MAPK. This triggers the cell proliferation and inhibits apoptosis. According to Callahan and Hervey 2011, about 25% of the breast cancer are classified as HER2 positive. HER2 positive breast cancer are caused by the overexpression of the HER2 receptor on a cell surface. When the HER2 receptors are overexpressed, this increases the survival and proliferation of the cell. Uncontrolled rate of the cell proliferation leads to the formation of tumor in breast cell.
The next part is biotechnological solution.
There are several therapies used in treatment of cancer. One of them is immunotherapy. One of the renowned medications used in immunotherapy is trastuzumab, or commonly known as Herceptin. Trastuzumab is a recombinant monoclonal antibody against HER2. Trastuzumab inhibits the proliferation of tumor cells that overexpress HER2. Trastuzumab can be administered alone or in combination with chemotherapy. Together with chemotherapy, trastuzumab reduces the rate of recurrence by 50% and increases the rate of survival by 30%. However, there is a disadvantage dealing with trastuzumab, which is the risk of cardiac complication along its usage.
Molecular mechanisms of trastuzumab are MAPK and PI3K at interference, antibody-dependent cellular cytotoxicity, and HER2 degradation. In MAPK and PI3K at interference, trastuzumab inhibits the pathway by interfering the dimerization of HER2. The binding between trastuzumab and HER2 will block tyrosine kinase SRC signaling. These processes will lead to the increase in cell cycle arrest and suppression of cell growth and proliferation of breast cancer cells. In antibody-dependent cellular cytotoxicity, trastuzumab attracts immune cells to tumor sites that overexpress HER2. For example, natural killer cells would target HER2 overexpressing cells caught with trastuzumab. In HER2 degradation, trastuzumab promotes the tyrosine kinase activity and triggers HER2 internalization and degradation. This causes growth signal cannot be received and inhibits the growth of breast cancer cells.
In conclusion, breast cancer is the most common cancer among women in Malaysia. 1 in 30 women will be diagnosed in their lifetime. HER2 pathway is one of the predominant signaling pathway that regulate normal mammary gland development and breast cancer stem cells function. The overexpression of HER2 receptor will lead to uncontrolled cell proliferation and inhibition of apoptosis, which cause the formation of tumor in breast cell. The biotechnological solution that can be used to fight breast cancer is immunotherapy. Trastuzumab is a recombinant monoclonal antibody that serves to hamper HER2 pathway by interfering PI3K, AKT, mTOR, and MAPK, ERK pathways, exhibiting antibody-dependent cellular cytotoxicity and triggering HER2 degradation. Thus, breast cancer can be compromised. Here is the reference, and thanks for watching!